So we just presented data from our phase 1 study looking at safety and feasibility of targeting CD123 with autologous CAR-T cells. And we've had, AML by itself is quite challenging, and unlike other heme malignancies which have been targeted with CAR-T cells, the success rates haven't been as great. So for instance, with patients with ALL or myeloma or lymphoma, there's been excellent responses and leading to FDA approval of these products. But with AML, on the other hand, it has been challenging, and there are several reasons. And one, of course, has been identifying a good target, which is expressed on AML blasts, but not on normal tissues. The other has been the complexity of the patients themselves, who are heavily pre-treated and still have poor outcomes and therefore have infectious complications and comorbidities. And lastly, the nature of the disease itself, which is very aggressive and rapidly progressive in the relapsed refractory setting, but also is immune suppressive by itself. Therefore, the cells that we infuse tend to have limited persistence and thereby overall poor response. So our approach has been to target CD123, which is highly expressed on AML blasts, but also on leukemic stem cells. And we developed a lentiviral construct that was actually generated in the lab of Dr. Paulina Velasquez. It creates cells which are a second-generation CAR that have a CD28 co-stim domain and also have a CD20 safety switch to ensure safety in case of toxicities. And our study is designed as a bridge to transplant approach, which therefore means that at the time of enrollment, patients must have an identified transplant donor. We've had thus far about 70 referrals to the study, which is quite a significant number, but of which we've been able to enroll about 20 patients and infuse 9 on study and 1 in a single patient protocol. Again, I think this highlights the challenge that we see with patients with AML, where their disease itself is very aggressive and rapidly progressing, which means the window is very small in which to treat these patients, but also their poor counts and infectious complications and comorbidities makes it very challenging to get onto these studies. And again, this is very different than our experience in ALL, which highlights really why AML has been difficult. We have 4 dose levels on the study so far, and we've treated on dose levels 1, 2, and 3. And so we're excited because we're seeing some evidence of expansion and also anti-leukemic activity starting with dose level 2. And indeed, we've seen 2 complete CRs, one with a patient with extramedullary disease and one patient who had a morphological CR. Although with low levels of MRD persistent. And this is especially exciting for us because these patients are very heavily pre-treated and have had really refractory disease. And except for one patient who had primary refractory disease, all the other 9 patients on our study have failed or relapsed following a bone marrow transplant, and either 2, 3, and even 4 transplants, which has been really showing the high-risk nature of these patients that we enrolled. And also, encouragingly, we have not seen any dose-limiting toxicities, and overall, the cells have been well tolerated. And we were really concerned with potential toxicities from CD123 targeting, including the risk of myeloid dysplasia, because these cell 123 is also expressed on normal hematopoietic cells, and also risk of endothelial toxicity, but we've really not seen any evidence of that so far. And I think going forward, we plan to keep enrolling on our higher dose levels, and also perhaps look at a donor-derived arm where we would manufacture T cells from the transplant donor, the previous transplant donor, to sort of overcome the issue of fitness of the starting T cell product for these patients.